Hello everyone, in this video we're going to talk about how molecules can be transported throughout a membrane, especially a cell membrane or any type of membrane inside a eukaryotic cell that's semi-permeable. Most of the membrane that we look at, well actually all of it, they're all semi-permeable membrane or selectively permeable membrane because cells and organelles need to regulate its internal environment. So right now we have molecule transport across membrane. How a molecule can transport across a membrane depend on certain properties of the molecule itself. So for example, if we have a really small molecule, such as oxygen, it can diffuse across the membrane directly, right? If we're talking about diffusion, that's high, going from high to low. If we have a small molecule, if we have this membrane right here, the small molecule can directly go from the high concentration of a small molecule to low concentration of small molecule going across the membrane. So that's the first case. If you have a larger molecule, for example, instead of going directly through the membrane, something called endocytosis or exocytosis would happen. And we'll go over that in the next video a little bit more. But basically, if you have, this is your cell membrane. If you have a large molecule that need to go out of the cell membrane, instead of going through directly because it's too large for the cell membrane, you would have a vesicle surrounding this molecule, and it will fuse and become a part of the cell membrane and allow that molecule to go out of the cell. Okay, one more time. So this is your cell membrane. You have a vesicle that has a molecule inside, and this vesicle would move to the cell membrane, open up, and allow your molecule to go outside. Same thing with the other, so this is exocytosis. Exo is going outside. Exocytosis is how a vesicle can send molecules out of the cell. And then you have endocytosis, where if this is your cell membrane and something's supposed to go inside the cell, your cell membrane will start pinching in, and then a vesicle will be formed from the cell membrane in order to deliver. So it will start pinching in and it will deliver the molecule inside your cell. That's the difference between endocytosis and exocytosis. Endocytosis, molecules coming in the cell, a vesicle is formed. Exocytosis, a vesicle already exists, but it becomes a part of the cell membrane to allow a molecule to go outside of the cell. The other one that we have is polarity of molecules. So if you have a nonpolar molecule, especially nonpolar small molecule, because your cell membrane is mostly nonpolar, then this nonpolar molecule is able to go through the cell membrane. However, you could also have a polar molecule that cannot go through the cell membrane directly, even if it's a small molecule, or it might go through the cell membrane very slowly because the cell membrane is mostly nonpolar. So for example, water is polar. Water can diffuse through a cell membrane. The word diffuse right here means you're not using energy and this molecule is going from high to low. That's the word diffuse. So water can diffuse through a cell membrane, but this process happens very slowly because water is polar instead of nonpolar. Okay? But if you have a polar molecule, a lot of times you would need a transport protein, a protein that's a part of your cell membrane, to allow your cell to allow this molecule to go through the cell membrane. So we'll look at examples of that. All right, this next one is called transport proteins. This next one is called transport protein. So when we said on the last slide, where if you have a really large molecule, then it's not going to be able to go through the cell membrane on its own, or if you have a polar molecule, it's not going to be able to go through the cell membrane very easily. Then you'll have something called a transport protein. So this transport protein can help with either diffusion, or in which case we call it facilitated diffusion, right? Facilitated diffusion means your organelle, sorry. So facilitated diffusion is talking about a molecule that can't just directly go through a cell membrane. It has to have a certain structure to help this process to happen. And in this case, if you have a protein that allows the organelle, if you have a protein that allows the molecule to go in one out of the cell, then we call that facilitated diffusion. There are two types of proteins that can do facilitated diffusion, or if you do active transport, we'll talk about that in a second. But there are two types of membrane proteins that allows molecules to go through the membrane. So one is called a channel protein, and one is called a carrier protein. So here is an example of a channel protein. This is an aquaporin that allows water to move through the cell membrane, and more quickly, because water can move through the cell membrane anyway, but aquaporin allows it to happen faster. So an aquaporin has a channel that completely goes through the two sides of the cell membrane, right? It's opening up to the inside of the cell and is open up to the outside of the cell. If you think about whether the inside of the channel will be hydrophobic or hydrophilic, it has to be hydrophilic because we're allowing water to go through. The next one is called a carrier protein. So instead of having a channel that's completely open up to both sides of the cell membrane, you have a carrier protein. The carrier protein opens up to one side of the cell, and then when the molecule that's supposed to be transported is bound to this protein, this carrier protein will go through something called a conformational change. So that's basically a change in the shape of the carrier protein. And then after this conformational change, instead of having this carrier protein open up to the outside of the cell, it would open to the inside of the cell instead, to allow this, in this case, glucose molecule to go from the outside of the cell to the inside of the cell. Okay, this is a channel protein where it's open up all the way through to the outside and the inside of the cell. And then we have here is a carrier protein where it opens up to one side of the cell first. When a molecule binds to the protein, it will go through a conformational change, a change in the shape, and this protein would open up to the other side of the cell. All right, passive transport versus active transport. Passive transport is high to low, but what are we talking about? High what? High solute concentration. Well, actually, never mind. High any kind of concentration. If you're talking about a passive transport of water, you're going from high water concentration to low water concentration. If we're talking about passive transport of a sodium molecule, sodium ion, you go from high sodium ion to low sodium ion. It doesn't matter what any of the other molecules are doing. When we talk about the transport, we're talking about that one type of molecule that you're looking at and how that's being transported. So if there's no energy required, and it's going from, the molecule is going from a high concentration of that molecule to a low concentration of that molecule, that would be a passive transport. If we have active transport, that's when we need energy, right? Because high to low, going from high to low is something that can naturally happen. If you spray Febreze on one side of the room, you can smell it on the other side of the room. There's nothing that you're doing to allow that to happen. It just happens because you have a high concentration of Febreze over here and no concentration of Febreze on the other side of the room, so it just flow over. However, active transport is against nature, right? Molecules don't want to go from a low concentration to a high concentration. So in order for that to happen, you have to have an input of energy in the form of ATP. Here's a word called concentration gradient. So when we talk about high to low or low to high, this is all talking about the concentration gradient. So a gradient is basically the difference, right? The steps. So if you're from high to low, we call it, we're following the concentration gradient. It's following the concentration gradient. And if you're going from low to high, we call it, you're going against the gradient. You're going against the current, kind of. So that's concentration gradient. The next one is population of molecules. Population of molecules. Maybe, I'm not sure what, I think this is wrong. Anyway, this one is more important. Dynamic equilibrium. Dynamic means something is changing. Equilibrium means there's no net change in concentration, right? Remember when we talk about equilibrium of a chemical reaction, at that equilibrium, there's still chemical reaction happening. And however, the forward reaction and the reverse reaction, I apologize for the leaf blower background noise, but we can get through it. So dynamic equilibrium is talking about equilibrium, which means you can have the same concentration on both sides of the membrane, right? And those concentration doesn't change, but there are still molecules going in and out of the membrane all the time. So that's what we're talking about. When we talk about membrane, the goal of doing a passive transport, for example, when passive transport happens, it's because molecules want to reach a dynamic equilibrium. But remember, the dynamic part is really important for equilibrium because it's not like the cell membrane is there and everything inside and outside of the cell are just sitting still and doing nothing. They're still going in and out all the time, except the concentration of that molecule, let's say sodium, inside and outside of the cell is all the same, and it will stay the same. So here's diffusion. We watched it in class. Here's diffusion again. Here's passive transport. We call it moving down the concentration gradient, right? Passive is moving down the concentration gradient, high to low. And then we have active transport, low to high. And osmosis. Osmosis is still high to low. But remember, whenever we're talking about high to low, low to high, we're only talking about one type of molecule. Because water moves a lot, obviously, and we talk about it a lot because it's really important for organisms, for biology. When we have a special name for the movement of water. So when water moves from a high water concentration to a low water concentration, we call that osmosis. But if you have high water to low water, that's the same thing as having low solute to high solute, right? So we're good on that. Last one, tonicity, is talking about hypertonic. Hypotonic, we'll talk about that in this PowerPoint instead. Okay, so this one right here, we're going to talk about solutions. With the solution, there's a word called homogeneous. Is really important. Homogeneous means everything is being combined completely, uniformly combined, right? So if you have a solution, you won't be able to separate out the solute and the solvent just through gravity, for example. You have to do something special to it. So for example, if you have salt water solution and you want to separate the salt from the water, you have to evaporate the water out in order to get the salt. Next one, a solution is made out of two parts, solute and solvent. How do you know whether something is solute and solvent? Obviously, if you dissolve something in water, usually the water is the solvent. However, what if you're looking at air right now? There are all those components in the air. How do you know which one is solvent and which one is solute? Whichever substance that you have the most amount of or the most volume of, we will call that the solvent in the solution. And what you have less of will be the solute. So a solute doesn't disappear. So when you have salt solution, the salt doesn't disappear even though you can't see it anymore. Here is if you were to separate a solute and solvent, one way you could do that is through distillation. Here's salt being separated from seawater. And what you also need to know is that a solution doesn't have to be a liquid. The solute could be a gas, it could be a liquid, it could be a solid. So for example, a steel is a mixture of iron and carbon, and because you have more of iron compared to carbon, you will consider steel as a solution, but iron will be your solvent. And for air, for example, we have mostly nitrogen, and the nitrogen will be the solvent to oxygen and all of the other gas. Here's diffusion. Diffusion happens high to low. We already said that. Diffusion as well. High concentration, area of high concentration of that substance, the area of low concentration of the other substance. Effect of temperature on diffusion. If you have higher temperature, molecules move faster. Diffusion happens also happens faster, right? Because if molecules are moving faster, they're bumping into each other and they're colliding, and whatever you're dissolving is going to be pulled apart and get mixed up in the water or whatever solvent you have a lot quicker. Then we have osmosis, which is water going from high water to low water. or low solute to high solute. That's the same idea, right? High water is low solute. No, high water is low solute. Um, Low water is high solute. So when you go, you have to go from high water to low water, or if you go from low solute to high solute. So in this picture, if we move this over, you can see that we have an area of high water concentration and an area of low water concentration through a selectively permeable membrane. Remember, biology is all about selectively permeable membranes. So in this case, this selectively permeable membrane only allows water to pass through, but not the sugar molecules. In this case, water is able to go from the high water concentration to low water concentration, and it could go from the low solute concentration to the high solute concentration. Either case, water will move from this side of the membrane to the left side of the membrane, eventually reaching dynamic equilibrium. Next one is called passive transport. We already talked about it. Passive transport is when you're moving down the concentration gradient, going from high concentration of that one molecule to low concentration of the other molecule, and it does not require energy. Active transport, the opposite of passive transport, going from low to high, and it does require energy. Next one, osmosis in the cell. You can see molecules moving through the cell membrane if it's a small cell. Oh no, if it's a small molecule, or if it's especially if it's a small nonpolar molecule, then they can go through like this. Oh, but right here we're looking at osmosis, so all of these are just water moving through. Here's a comparison chart between osmosis and diffusion that you can look over on your own. Now, the tonicity. There are three types of tonicity. There's isotonic, hypertonic, and hypotonic. Isotonic means the same. So whatever you're thinking, you can think about solute, you can think about solvent, you can think about water. Whatever it is, everything is the same, right? So if you look at this picture right here, we have the same. Well, when we talk about the same or high to or low, we're not talking about the amount. We're talking about the concentration. Always remember, we're not talking about the amount. We're talking about the concentration. It's the concentration that matters. So right here, you have the same concentration of solute on both sides of the cell membrane. This is a cell membrane right here. You have the same concentration of solvent on both sides of the cell membrane as well. Next one, hypertonic is when one side of the cell membrane has hyper means more, but more what? When we're talking about hypertonic or hypotonic, it's always the solute. So hyper is high amount, but high amount of what? High amount of a specific solute that you're looking at, okay? So right here we have a hypertonic solution. If we're comparing B to A, it's kind of like you can't say a person is short unless you're comparing this person to another person, right? Now, there's nothing wrong with being short, but you can't say something is, large or small unless you're comparing it to something, because it's all relative. Same thing with solution. For hypertonic, it's a hypertonic solution compared to this other side. And then in this case, a hypotonic solution right here is hypotonic compared to this other side. For a hypertonic solution, water will move from an area of low concentration of solute to a high concentration of solute, right? If you just look at this picture right now, you can always draw a picture like this for yourself to help you answer the questions. High to low, high water to low water, or low solute to high solute. That's how water moves. And if you were to put a cell into a hypertonic solution, then there's more solute on the outside, and then your cell would shrink. If you were to have a hypotonic solution, such as the A side, then water is going to be lost from the A side, because hypo is low, low solute. Low solute, the water needs to move away from it. That's hypotonic solution. If you were to put a cell into a hypotonic solution, the water will move into the cell. Let's say you put a cell into water, for example. The water will move into the cell, and then your cell would expand or even burst open. So that's not good. We'll go over this a little more in a little bit or in the next video.